All right, well, it's a rainy day in Bethesda, so our attendance is down, but that's why we videocast, so everyone can watch it on the web. So we're pleased today to have Pete Choyke. He got his MD from Jefferson Medical College. He did a residency at Yale New Haven Hospital, and he did a imaging fellowship at the University of Pennsylvania. He joined the faculty at Georgetown. I was at GW, nearby. And he came here and started the molecular imaging program. So his title today, Imaging of Cancer. Pete.
Thank you, Terry, and good afternoon, everyone. What I'm going to do today in this talk is explain the various tools that are used to image cancer in a radiology department, a modern radiology department. And we'll go over a little bit of the engineering and physics behind some of the machines and what they can tell you and what some of the limitations are behind the machines. So, of course, I'm not going to dwell very much on why we do imaging. It is pretty self-evident. It's a key element of screening. And I guess I'm going to step out here if it's okay. Detection. So is there a tumor at all in a body? And then staging, which is a very important characterization of the tumor, how far it's spread outside within the body as it metastasizes, which has impact on how it's treated. And then once all that has occurred, patients go on treatment, and we look to see whether they're better or worse, so-called monitoring of treatment. If the disease goes away, we monitor patients for recurrence as it comes back. And ultimately, we often ask the question, what do we think is going to happen with a particular patient given the constellation of findings that we see on imaging? So, prognosis. So as you walk into a modern radiology department today, the key tools are computed tomography, MRI, ultrasound, single photon emission computed tomography, and positron emission tomography. 50 years ago, when you walked into a radiology department, there was an X-ray machine, maybe a fluoroscope that did real-time X-rays, but there really wasn't much information in those images. It's been completely revolutionized over the last 20 or 30 years by these cross-sectional imaging techniques that show the anatomy. Now, this is sort of a joking slide about name that scanner. It's really very difficult, unless you're an expert, to differentiate the way the scanners look as you, say, walk into a room. They all have the gantries and then a bed where the patient sits. The exception is the ultrasound machine, where you actually do have a chance at identifying. Those look completely different. They don't have those gantries, at least not yet. So these are the various machines. So let's talk about CT, computed tomography. Don't call it CAT scan. If you want to be cool, call it a CT. This is a cross-sectional technique that depends on X-ray attenuation of tissue. Here's a lung cancer that's caused a pneumonia on the left side. By the way, all of these images have the right side on your left and the left side on your right, as if you were looking from the patient's toes toward the head. And that's a convention that's universal across all the modalities that I'm going to talk about. So now this is in lung cancer in the left side. It has a pneumonia. It also has extension to the hilum. So we learn a lot just from this single image. Now, CT has a lot of advantages, and it's the reason why it is the workhorse of the radiology department. First of all, it's widely available. It requires virtually no preparation on the side of the patient, on behalf of the patient. except perhaps to drink some fluid beforehand, maybe 15 minutes to an hour beforehand. The patient will drink some fluid to opacify their bowel, although that's some changing with the modern machines. We're getting even away from that. It's very rapid. Sort of conventionally, a scan takes from the neck to the pelvis, 30 seconds, but our new machines in the Department of Radiology do that in less than 5 seconds. In fact, the table moves so fast that they have to put a brake on the table to slow it down gently so that the patient doesn't jolt off the table. That's how fast the machine is moving. So this is very, very quick, and it's very high resolution, as that image that I showed you. And nothing is cheap in the radiology department, but relatively, it's inexpensive. So it has many advantages. The disadvantages are that it involves radiation. Now, you might think that's paradoxical in talking about cancer. The patients already have cancer, so why are we concerned about radiation? We'll talk a little bit more about radiation. It's become a very important topic in the media. Patients are very concerned about radiation, and the whole industry of CT is moving very quickly to reduce the amount of dose that a patient receives. And it turns out, because we haven't been paying much attention to this topic over the years, that there's actually a lot you can do to reduce radiation without changing the accuracy of CT by factors of 2 and 3 reductions in radiation without any change in efficacy. So it's very important. Another disadvantage is that it sometimes requires intravenous contrast media, and we'll talk a little bit about that. And it only provides anatomic information. There's no physiologic information coming from a CT scan. It tells you that there's a blob here, it tells you there are normal structures there, but it doesn't tell you anything about the function that's going on. So might as well start with how we get a CT scan. And it all begins with the cathode ray tube. So this device is over 100 years old. Wilhelm Röntgen sort of came up with this idea, but it's been very refined over the years. And what happens here is that we have a coil that burns off electrons in a magnetic field that sucks the electrons toward an anode, which is the target. And so these electrons accelerate towards this target, hit the target, and produce X-rays. And that's what's coming off. Now, by tilting this, you can actually get the X-rays to go in a certain direction. And of course, that's important if you're trying to make an image. You don't want them flying all over the place. You want them to go down in a particular direction. So we incorporate one of these X-ray tubes in the CT gantry, and it rolls around the patient circularly. And there are detectors. There used to be film, but now these are all solid-state detectors that pick up the X-ray, convert the X-ray into a little bit of light, and then that light is detected. And that's the way we begin to gather the information for the image. And then the patient rolls through this on the table so that you scan serial axial sections of the patient. Now, how an actual image is formed from a CT is a little bit complicated and really beyond what I want to talk to you about today. But you should understand something about it because the fundamentals are not that hard. And it really, if you think about 4 samples in the center of a circle, A, B, C, and D, and you just imagine that you shine light in various directions. Forget that it's an X-ray. Just shine light. You know that those objects will cast shadows, much as this image depicts. And if you keep iterating multiple times, you'll start to build up an image that reflects the location of these samples. Now, if these samples have X-ray attenuation, that is, they're dense to X-rays, the same thing happens. It's basically just shadows building up. And eventually, if you take enough views, you'll get a pretty refined image. The image that you see is very crude, of course. You couldn't do much with that. But if you do that 500 times, you might be able to really improve the image. And if you throw in some image processing techniques and something called Fourier transforms, you can really improve the way the image appears. That's all I want to say about it, but you can sort of understand the idea of back projection as the way to form an image. So the modern scanner has these X-ray sources that are rotating rapidly, and it creates this X-ray fan beam that goes into the patient and is detected by these detector arrays. And then there are these built-in data acquisition systems that right at the point of source of where the X-rays come in, they gather the information extremely quickly and send it out to the control room. Now, you might have heard the term spiral CT or helical CT. Actually, that term is kind of going away as it becomes so standard. But it used to be that we would do one full rotation, then move the patient, and do another full rotation, move the patient, do another one, and then collect the stat. That's not the way it goes anymore. Right now, the patient moves as the X-ray beam moves, so that it shows the pathway of the X-ray is actually a spiral around the patient. And so the information gathered is not slice by slice, but it's the whole volume of the patient is gathered at one go. So it's a volumetric acquisition, and sometimes known as a spiral CT. So what's the advantage of doing these kind of volume CT scans? Well, the native image is this image in the axial plane. And this is a little bit of the liver, here are the kidneys, the vertebral body, etc. But because it's a volumetric acquisition, you can then reformat the image to be coronal or sagittal. The sagittal is particularly useful for looking at the spine. It's very hard to look at the spine in this plane, but it's simple to look at it in this plane. And so this multiplanar ability really brings huge advantages to CT, to the modern volumetric CT, as opposed to the old CT where that would look very blocky or steppy if you tried to just stack images that were obtained at slightly different times. So one of the cool things that you can do now on the fly is change the intensity of the X-ray beam, depending on how much tissue it has to go through. So for instance, as you go through the lungs, there's much less attenuation than if you go through the abdomen, where there's a lot more stuff, and therefore X-ray attenuation. So you can sort of lower the strength of the X-ray as it goes through the chest, and then as you sense that it's going into the abdomen, you can increase the dose. And that way the patient gets a lower dose. So that brings us to this topic of radiation. And what the modern scanners can do is greatly reduce the dose that the patient experiences. And this is just a chart from MGH where they show, just by paying attention to some very simple things, they were able to reduce the radiation, which is measured in millisieverts, from, this is before and after they implemented some of the changes. So some of the things that you can do to make the CT scan more efficient, that is, lower the amount of X-ray that you need to deposit it, is to lower the kilovoltage of the X-rays. We were stuck at sort of 140 kilovolts for a long time, and then we began to realize you don't really need to give all that high energy you can go to lower energy. So some of the scanners have reduced it to 120 or 100, and so you get gains there. The detectors that I talked about become more sensitive, so you don't need as many X-rays to form an image. There are better reconstruction algorithms than the sort of the one that I described, back projection, where you take all these images over and over and over and over again. Well, there are more clever reconstruction algorithms that sort of shortcut that, fill in data, even where it doesn't exist, but based on the prior data, you can figure out ways to just put data in the scanner, and it allows you to make beautiful images without actually increasing the dose. And then there are images that are so-called synthetic images. That is, based on one scan, you can sort of compute another scan. And that's used particularly for patients who receive intravenous contrast. You can do a synthetic image that looks like there's no intravenous contrast. So you don't actually have to acquire that. You can calculate it from the first image. So that is very cool, and the reductions have been dramatic. So here's what I mean by the virtual image, or the synthetic image. So this is an image that was obtained on a regular scanner with intravenous contrast, and this was actually acquired. And you can see the kidneys and liver, and here's a cyst. And this is an image without the intravenous contrast, but it was obtained synthetically. It was calculated by taking out the calculation of what the iodine in the contrast media had, and then taking that out electronically, leaving you only with this image. So that sort of brings us to this topic of this iodinated contrast. This is the contrast agent that's injected at the time of a CT to opacify the vessels, and because tumors are vascular, tumors will light up as well. So very commonly, if you walk into a CT scanner suite, you'll see the gantry and the table, and this device that's standing right next to it, which is the intravenous injector or pump. And so this iodinated contrast is loaded up into a syringe and then injected into the patient pretty rapidly, 3-4 cc's per second into the patient. That's a very rapid injection, so you can get a lot of iodine very quickly, and that picks up on the CT scan. Now, so that's what this looks like. So there's basically one of these syringes is for the contrast, and the other is for a flush, to flush normal saline through the line after the injection. And this is just what it looks like. Now, the contrast itself, there are various manufacturers. This is just one that we use, and they're called non-ionic contrast agents. So in the old days, there would be burning and feelings of heat, but that's really much, much less now with these modern non-ionic iodinated contrasts. So just as an example, this is IsoView, and it has, it's a regular, benzene type chain, but there are at least 3 iodines attached to it per molecule, so that you can get a fairly high density. Iodine will block X-rays, so you'll see exactly where the contrast has shown up because of the presence of iodine. Now, for all its virtues, CT is not that sensitive a technique. And as we'll talk about some of the other modalities like SPECT and PET, this actually requires a lot of dose. So 30 to 45 grams of iodine are administered in a dose. It's quite a lot. Unfortunately, if your renal function is normal, that's excreted very rapidly. But if there is renal dysfunction, then that could be an issue. That's quite a bit of iodine. So I want to go over sort of the basics of looking at a CT scan. These 2 images are exactly the same, but they look very different. So on the left is a so-called mediastinal window, where we accentuate structures in the mediastinum, like the heart and the aorta here. There's a nodule here. And that's fine. Just by changing the windowing on the image, you can get an entirely different appearing image, so-called lung window, where you can now see structures within the lung, like blood vessels within the lung. You can still see the nodule very well, but these other vessels. And you can actually see very tiny nodules in the lung using this kind of technique. So same image, just windowed differently. If you will, it's kind of like photoshopping, where you don't change the actual image, but you change the parameters and you can bring out certain aspects of it. But I don't like the implication of photoshopping that you're actually changing the image. So I avoid that. But, you know, so really you get, for every image, 2 or 3 different windows that give you access to different kind of information on the scan. So here, the soft tissues, here, the lungs, and in another window, you could actually accentuate the bones. And there are even others that can be used. So it's a very flexible technique. And it's all based on this kind of grayscale where you set the so-called window, which is the length of the grayscale, and then the level, which would be the center of the grayscale. And by changing the center and the level, you can alter the way the image looks. Okay, with that, I'd like to move on to magnetic resonance imaging. And here's a prostate cancer on the anterior surface of the prostate. Here in the pelvis, here are the hips and some of the pelvic bones. Here's the rectum and some fat around that. And this is the specimen from the patient showing the prostatectomy and the cancer as depicted by the MRI. So it's a very powerful technique, and it's used in a variety of ways. So what are the advantages of doing an MRI over, say, a CT? Well, first of all, there's no radiation. It works on magnetic fields and radio frequency. It is multiplanar inherently, so you can actually acquire the images in a particular plane, which has less advantages now that CT is obtained in a volume, and you can cut it in any plane, but it's still advantageous. And most particularly, MR has multiple contrast types. CT only has one, how dense it is on X-ray. MRI, you can look at the T1 relaxation characteristics, the T2, the diffusion weighting, how it enhances on contrast enhancement, and spectroscopy. So a lot of different things working. Disadvantages. It is slower than CT. Remember we said we were up to 3 to 5 seconds through the whole body for a CT? Well, that's more like a half an hour for an MRI. It is more expensive related to how fast it is. It doesn't depict calcifications at all, so that limits it in some applications. And there are also some safety issues related to the magnetic field. This is a very strong magnetic field in a hospital environment. So ordinary objects, like scissors, become projectiles in an MR. So there's a big safety issue around having an MR in a hospital. There are zones of preparedness, if you will, where no objects that are magnetic can be brought into the certain area, and it has to be carefully monitored. It's also incompatible with some metallic implanted devices like pacemakers and cochlear implants, among others. Although that's changing. The pacemaker manufacturers are figuring out ways to make their pacemakers still compatible with MR. Again, we're not going to be able to talk about all the physics of MR in a talk like this. Not my intention. But I do want to give you sort of a gestalt or a feel for how MR works. So in our body is lots of water, and the water is made of H2O, 2 atoms of hydrogen for every molecule of water. And in fact, those little hydrogens are like little magnets. Now, we don't really think about them because they're spinning like little magnets, like little tops in your body. But since you're not really in a magnetic field, except for the Earth, there's no real orientation to those protons. But as soon as you apply an external magnetic field of some strength, you can get these guys to align, like little soldiers, straight with or opposed to the magnetic field. So now you can do something. Now they're all kind of organized. And if you come in with a radio frequency that is at the so-called resonant frequency of those little spinning protons, you can get them to do tricks. Like, instead of being aligned with the magnetic field, you can get them to be aligned perpendicular to the magnetic field. That's cool. Or even anti-perpendicular, you know, so completely opposed to the main magnetic field. Then when you turn that the RF off, you remove that energy, they start to relax back to their native state, aligned with the magnetic field. Well, when they do that, they give off energy. And the energy can be detected by a little radio receiver depicted here as this little C that will pick up the little signal. And then you can start generating an image from that. So basically, you have all these spins that are aligned with the magnetic field. You come in with the radio frequency pulse and divert them from their native state, which is aligned, to not aligned. And then as they recover, you get the signal back. And you can get these very complex pulse sequence diagrams that I won't go into. But the bottom line is, by applying a combination of radio frequency pulses and magnetic gradients, you can define where and how you're going to image a particular object. So very much like CT, there is the concept of back projection. And so if you take those four objects like we had before, A, B, C, D, and you apply the magnetic field, that's the big magnet, then you apply the RF and gradients, you can sort of back project images of these four objects. And extrapolating to a whole body, you can see how very complicated, but you could generate an image. So let's look at the anatomy of an MRI scanner. These are those big scanners most of you have probably seen or have actually had an MRI. And what's going on in that machine? Well, in order to generate the large magnetic field, you have to apply a large amount of energy inside the magnet. And to keep that going, you need to keep it at liquid helium temperature so that it's superconducting. So you charge up the magnet at high energy, and then you keep it going by cooling it down to liquid helium levels. So we have something called a cold head, a refrigerator compressor, and then this liquid helium that wraps around the actual coils to keep them very cold. And then the patient, of course, goes in here. The magnetic field is generated by this superconducting wire that has a lot of current going through it. So as we look at this magnet in a typical room, we have our friendly injector, just like in CT, two barrels, one for the contrast agent, one for the flush. You have the scanner itself, and the magnet is in here. Now, one thing you'll notice is coming out of the top of that is this manifold, and that's for the so-called quench pipe. And what's that about? Well, it turns out just what I described is great until something goes wrong with one of those wires, and there's a little short, and that heats up very rapidly, and that heats up the liquid helium into gas, and that gas will just come out of the magnet very quickly. In fact, so quickly that it will replace, to a large extent, the oxygen or air in the room. So we have a problem. If the magnet quenches, that is, the superconduction goes away rapidly, we fill up this room with helium. If there's a patient in the scanner, they can't breathe. So in order to avoid that, we have something called a quench pipe that's out of the top of the magnet, and it will vent the helium out of the building, or out of the, at least out of the room. Very important for that quench pipe to work when it's needed. I've only seen it happen a couple times, one purposely and one not purposely, but it's very dramatic when it happens. Things happen very, very quickly. So the other thing I put on this diagram about safety is ear protection, because this thing is very loud. I don't know if any of you have had an MRI, but they're very loud, and the tech should put often two forms of ear protection, earplugs and then these headphones, and we need to protect the patient's hearing. So I mentioned the problem that in a strong magnetic field, ordinary objects like scissors or oxygen tanks become projectiles and can actually hurt people who are in the room. So while this is kind of a joke, in fact, this is very, very serious. So MR scanners are very powerful. Objects are attracted to them and easily reach 60 miles an hour by the time you let go to the time it hits the gantry, which is only 10 or so feet. And a sharp or heavy object can be deadly to anybody in the path of something going 60 miles an hour. So the technologists who run these scanners are absolutely trained to make sure patients don't walk in with anything metallic, healthcare workers don't walk in with anything metallic, and even doctors don't. And if you're not careful, stuff like this happens, like a buffer machine, because they were cleaning the room and they didn't realize that the magnet was on. The magnet is always on. Doesn't matter whether it's midnight, 3 o'clock in the morning, or 3 o'clock in the afternoon. The magnet's always on. And these poor people even thought about the idea of trying to restrain this cabinet. You see, they put ropes around it, but it still got sucked in to the magnet. So it's very strong, and we take this stuff really seriously. And unfortunately, a few people have actually been killed by especially oxygen tanks, because you can imagine they're like torpedoes. And so we have warnings all over. We have a lot of training. The hospital makes the risk management in the hospital spends a lot of time on MRI. We have 20 MRI scanners in this hospital now, and you can imagine, you have to maintain quality on each one of them. So let's talk a little bit about MR contrast agents. Here's an MR contrast agent showing us a tumor in the liver that would be ordinarily hard to find. So the contrast agent, rather than iodine, is gadolinium. Gadolinium is so-called paramagnetic. That means it influences the rate at which the protons relax back to their native state. They relax back faster when gadolinium is around. And there are a lot of different companies with different chemical structures in their contrast agents. So these are the names of the agents that are available, but I want to talk about one aspect of them, and that's the thermodynamic stability constant. And you may say, well, that's a little esoteric. He's talking about imaging. What do we care about thermodynamic stability constants? Well, these are log numbers, and so you can see that this agent, what we call gadolinium DOTA, has very excellent, very high thermodynamic stability, and most of the agents that are approved have good ones, but there are a couple at the top that have much lower stability, which means that the gadolinium ion falls off more easily. Now, for most patients, that doesn't make any difference at all, because if it falls off, it gets peed out in the urine very quickly, and there's no harm, no foul. But for patients with impaired renal function, that gadolinium ion may stick around quite a bit. And that unfortunately resulted in a disease entity that was reported in the late 2000s called nephrogenic, for renal failure, systemic sclerosis. And it caused a fibrosis in the tissues that was very, very debilitating, permanent, and very problematic. So just a few words about that. It was discovered really in Denmark in May 2006. By November 2006, others were beginning to report that this happened with gadolinium contrast agents only in patients with impaired renal function. And about 200 cases have been reported. But since 2013, once we identified that this was a risk factor, there have been no cases reported. So, we've identified why this happens. We don't give the contrast agents like Omniscan or Optimark generally at all, but certainly not in patients who have impaired renal function. And so we've essentially eliminated this, but you will see there's a lot of concern about the renal function of patients when they undergo an MRI. Why? Because of this. You've got to know that the kidneys can clear it. So these are some scans of a patient who unfortunately developed NSF. So gadolinium is highly toxic by itself, but it's in the form of a contrast agent, it's chelated. So the body just doesn't see it, doesn't have access to the ion, and it's excreted into the urine. But patients with abnormal renal function, that chelates, circulates, and circulates, and circulates over days, and eventually, if the thermodynamic stability isn't very good, it falls off that molecule and becomes free ion that deposits in soft tissues. So the risk factors are renal failure, the dose that you give, what kind of contrast agent, and of course in this situation you should always think about either not using MR or using some other form of imaging. So let's talk about ultrasound. Ultrasound, most people are familiar with because of obstetrical ultrasound. It's got some great advantages. It's no radiation, it's real time, it's inexpensive, it's quick, and doesn't require injection, although they're desperately trying to develop contrast agents, so that advantage will go away. But for now, usually no injection. The disadvantage is that it is very operator dependent. You have to be skilled in ultrasound to get a good image. It's not something, it took me a year to figure out how to do ultrasound successfully. The other big problem is it's kind of like a flashlight. Now, I analogize CT and MRI to like turning on the lights in a room. You can see everything in the room. It's no big deal. Imagine that you were trying to figure out what was in this room with a handheld flashlight. That's more like ultrasound. If you don't point it at the object that you really want to know about, you won't see it. So that's why the skill comes up. So what you see is all there is. And it's difficult to quantify because it's just sound waves. And there are just some tissues that won't permit the transmission of ultrasound waves, like the lungs, air. Sound just bounces right off. So you can't image the lungs. You can't image the bones. And because of that, you can't image the brain because that's surrounded by bone. And there are other examples. So there are some limitations. So what happens here? This is really kind of simple. You have a sound generator. It sends waves into the tissue. They meet an object. They reflect off that object, and they're detected. Ultrasound. Now, lots of things can happen when you put a sound wave into a body. The sound wave can get attenuated. It can get absorbed. It can get reflected. It can get scattered, refracted, diffracted. And so you can get kind of a complex relationship between the signal that you send out and the signal you receive. So here's a patient with multiple small metastases in their liver. And they're very subtle on the ultrasound, but if you were able to see the real-time image, they would be much more apparent. So we have probes that the technologist holds, and they are the ones that produce the sound through a piezoelectric crystal, sends the sound waves out, and they come in all kinds of different shapes and sizes in order to accommodate different frequencies. So the higher the frequency, the better the resolution. So if you want really good resolution, use fairly high frequency probe. But the problem is that sound attenuates as a function of frequency. So the problem with a high resolution probe is that you can't go very deep. So it's great for the thyroid gland and for what they call small part scanning, but it's not very good for looking at the abdomen, where you actually have to have quite a bit of depth. Now, one of the things that's evolving rapidly in ultrasound is the size. So Most of the machines we use in the hospital are these high-end machines that are kind of big and heavy, and you know, you can roll them, but they're not easy to roll. But more and more, we're seeing scanners that are handheld or even can be applied to part of something like an iPhone. And in fact, there's an iPhone attachment that allows you to do ultrasound with your iPhone. The image quality today is not as good as what you can get with the very high-end machine. But I have no doubt that in not too many years, the image quality will be excellent at these small sizes. So there's no reason why physicians can't walk around the hospital with something this size and apply ultrasound at the bedside in the future. People have been talking about the new stethoscope. That may be the new stethoscope. And another thing that's really great about ultrasound is because it's real time, you can do things like biopsies very easily. So you put the ultrasound probe on the patient's skin, and then you can see the lesion, and you can see the needle as you put the needle in to hit this particular lesion. And because it's real time, you can watch that and monitor your direction and all that. So it's really a great way to do biopsies. So as I mentioned, you know, every one of the modalities I'm going to talk about has its own contrast agent. The contrast agent for ultrasound is called an ultrasound microbubble. So it's just like those bubbles that the kids blow, you know, and make bubbles. That's exactly what these are. But they're filled, instead of with air, these are filled with perfluorocarbons. And that just keeps them a little bit more stable. So when the sound wave comes, they reflect the sound very strongly. So here's before the contrast is given, and here's after the contrast is given. You can see these lesions just lighting up really well because the sound is bouncing off these ultra small bubbles. What's kind of interesting about them is if you apply more sound, more higher energy sound, you can actually burst them. And that becomes useful in a couple ways. First, if you want to figure out what the blood flow is to something, you can burst all the bubbles and then watch the inflow of the new bubbles. And that gives you some indication about how fast the blood is flowing into something. So that's one good thing. The other thing is you can actually encapsulate something like a drug into a microbubble. And so when you see the microbubbles at the target, you can destroy the microbubbles, releasing the drug right there. So that's kind of a neat application. So let's go on. I'm going to move on to, in the last 15 minutes, to talk about single photon emission computed tomography, which is called SPECT, and PET. So single photon emission is combined with computed tomography to make SPECT. What's single photon emission? Well, it's kind of a fancy word for radioactivity because it's a isotope that gives either an alpha ray off, a beta ray, or more particularly a gamma ray. That's what we're looking for. And those gamma rays then go through the tissue and create the image. So I'm going to put the SPECT advantages and disadvantages on one slide. It's relatively inexpensive, and there's a broad experience and distribution of machines along the country. Most of the applications are in cardiac, not in cancer, but there's still a lot in cancer. The disadvantages, of course, it involves radiation. It involves some kind of preparation of this radioactive imaging agent. There's a lot of regulatory surrounding that, as there should be, and scanning is much slower. So what needs to happen is you have a radioactive isotope, and that's attached to a compound which targets cancer in some way. Now, one of the simplest examples is the bone scan, which is technetium-99m. So that's a radioactive isotope derived from molybdenum. And that's attached to methyl diphosphonate, which binds to hydroxyapatite in bones. So wherever there's a lot of bone turnover, as in a bone metastasis, this agent will take up. So here you have a patient with prostate cancer and a lot of bony metastases. But technetium only lasts so long. It has a half-life of 6 hours. So that means you only have a limited amount of time to image the patient after, and by 24 hours later, there's nothing left. You put this patient on the scanner at 24 hours, you won't see anything. It's gone. So there's a timing issue. So there are a number of agents that are used for cancer. The bone scan that I just mentioned, the pertechnetate scan, which also uses technetium, is used for thyroid and the salivary gland. Thallium is used for the parathyroid. Then there's indium, which has a really long half-life. That's about 70-something hours. And that's used to label white blood cells and send them in and see where they take up. That can be used in lymphoma, but also in infection imaging. And then iodine-131, which is taken up by the thyroid, but is not only a imaging method, but also a treatment method. So this is a patient who had cancer of the thyroid. I-131 was given, and then on return, the patient has much less activity in their thyroid because the I-131 has killed the cancer. So I want to say a few words about this because it'll make more sense when I talk about PET. Back projection is the same basic principle. We're not doing anything fancy here. But there is this problem that when you inject something that's a single photon emitter into a patient, there are emissions that go on in every direction possible. Sort of a cloud of gamma rays come out in all directions. So how do you possibly make an image out of that? Well, to do that, what you really need to do is to force some of these gamma rays through a channel. So at least you have some idea where they're coming from, and that's called a collimator. So you run it through this collimator, and then there are the crystals that detect the activity, and then you can form an image off that. So this is what a collimator looks like. It's actually often made out of tungsten, and they're deep holes, and you can see that a gamma ray has to go down one of those tunnels to get to the detector. If it doesn't go through there, if it's at an angle, it'll be blocked. It won't get through. So what's the implication of that? That means that many of the events that happen, we talk in terms of events in radioactivity, many of them don't get captured. They happen, the patient's exposed to the radiation, but you don't capture it on the image. That's not good. And it makes SPECT relatively inefficient because there's a high rejection of events. So this is just another way of showing the same thing. You have this source, you put it through a collimator, and only these gamma rays that are sort of parallel to the collimator will get through to the detector. So it's inefficient. So this is what a SPECT camera looks like. It's generally combined with a CT. So you'll have these two flat plates that will rotate around the patient. These are the gamma detectors with the collimators on them. And then they roll around, and then you get a CT scan at the end. So then you can superimpose the activity from the radioactivity on top of the CT scan. So you get localization of where the activity is. So it's just superimposing the two images on top of it. So radioactivity is a problem. We've already talked about it a little bit. It's a problem for the operator who every day has to administer radioactive material. It gets a dose to their hands. We all walk around with these radiation badges, rings, and things like that. And then to the patient who doesn't do this very often, but nonetheless gets a dose from the CT. Okay, so let's, in the final minutes, talk about perhaps the most high end of all the imaging that we do in an imaging department, and that's positron emission tomography. So very briefly, a positron is a positive electron. And if you like to be kind of avant-garde about it, it's an antimatter. Because when a positron encounters an electron, they annihilate each other and form two gamma rays in opposite directions. So you can see there's some advantages compared to single photon emission. Single photon sends them off in any random direction. You got to collimate them to collect them. But here we have this nice little setup where you get 2 gamma rays for every event, and they're in opposite direction. So if you detect one at one side and the other at the other side of the ring, well, you have some fair idea where they came from. They came from somewhere along that line that, where the event occurred, which is way more information than you get with a SPECT. You have no idea where that event came from. So that has good advantages. It's highly sensitive. It's the most sensitive technique we have. Whereas we talked about grams of iodine for CT, we talk about picomolar concentrations for PET. And that means that you can do things like receptor imaging, cellular receptor imaging. You can get metabolic information. There's much more spatial resolution than SPECT because you know where these events are occurring. And of course, you can combine it with CT so you get the best of both worlds with high accuracy, high sensitivity. There are some disadvantages, as always. Expense. You can imagine this is a complex technique. Regulatory, as always, and the half-life of the agents. So the PET gamma rays are always 511 keV photons, and they're always produced by this positron annihilation. And they come out at 180 degrees. So just to reiterate, so this is F18, which is one of our favorite PET emitters. It will emit a positron from the nucleus, and because it has some energy, it'll go away from the nucleus a certain distance, several millimeters actually, before it encounters its electron. And it's not really love at first sight because they annihilate each other. And then 2 gamma rays get formed, and those are detected at opposite sides of the detector. So one of the best agents, and amazingly one of the first, and actually the NIH plays an important role in this story, is the agent called F18 fluorodeoxyglucose, FDG, fluorodeoxyglucose. And the reason why that's important is this guy, Otto Warburg, who in the 1920s figured out that cancers turn off aerobic glycolysis, this Krebs cycle, and preferentially use anaerobic glycolysis or aerobic glycolysis, where pyruvate is converted to lactate. So they draw in a lot of glucose into the cell, but instead of sending them through the Krebs cycle, they send it to lactate. And that's the way cancers generally create energy. So with this tracer, F18 FDG, you can do a lot with imaging cancers. So basically you inject it into the bloodstream, it escapes from the blood vessels, gets into the cell, and comes in through the glucose transporter, just like glucose. So the cell doesn't know that it's got an F18 on it. Glucose comes, and then it's catalyzed by hexokinase. But the hexokinase recognizes that there's something wrong with this glucose. It doesn't let it go on to pyruvate and to lactate. There's something funky about it. It's got this deoxy thing on it, and it's got fluorine in it, and the body says, I don't know what you are, but I'm just keeping you in the cell. And so it accumulates in the cells that have abnormal metabolism. So you can get uptake in cancer. So this is a patient with a colon cancer who's been operated on. This is the bladder and the Foley catheter. Here's a part of the kidney. And this is the very small tumor. You could never detect this with any other modality but FDG. So it's just this very small glucose-like molecule that's utilized. So that is most of what we do on PET is with FDG. And all of these scanners have a PET component, have a CT component, and then the fusion component where they fuse the PET and the CT. So this is kind of what, this is a breast cancer patient who has a lot of activity here in the anterior mediastinum. in the bone. And this is what the PET scan itself looks like, but you can see how much better it is to be able to localize here in the bone where the activity is going. So you can really get the best of both worlds with PET and CT. So just a few words about the vocabulary. I'm going to finish up in just a second. We have the emission scan, which is the way the activity is emitted by the patient. And then there's a transmission scan, which is an attenuation map of the patient. And that relates to how much CT attenuation there is. And the machine compensates for that, so that something that would block, like the bone, which would block a lot of X-ray, is compensated for, so the activity isn't abnormally reduced in the bone. So if you don't attenuate the PET scan, you get kind of crazy-looking images. But after attenuation, you can really see where the pathology is trusted much more. So PET has the advantages of being very high energy, high sensitivity, and moderate specificity, the ability to correct for attenuation, no need for collimation, and pretty good resolution. So I've taken you through a radiology department, a modern radiology department, and we'll just go through a brief summary. So as we look at resolution, the top of the heap is CT, followed by MRI, followed by ultrasound, followed by PET and SPECT. So if it's resolution you need to see something really small. Generally, CT is the best. But in terms of sensitivity, what small thing can you detect? Not physically small, but low in concentration. For instance, a receptor on a cancer cell, or a small amount of glucose being taken up by a cell, a cancer cell. PET is at the top of that pyramid, followed by SPECT, followed by ultrasound with microbubble, as it turns out. MRI and CT is at the bottom of the sensitivity scale. So you can see why PET and CT are kind of complementary of each other. One has great spatial resolution, the other has great sensitivity. Now, what about cost? Well, this is really important, especially as we enter a more cost-conscious world, but also as we look at developing countries. By far, the most effective imaging, cost-effective imaging, is ultrasound. Handheld, small device, fits on your iPhone. And so in the third world, this is the agent, this is the imaging technique of choice. And then followed by these others, with PET clearly at the top. So overall, the workhorse is CT. Specialty cancers like brain and liver and prostate, MRI is the first choice. For problem solving, cyst versus ultrasound versus solid, ultrasound is the technique. For looking at bone metastases, we use SPECT-type scanners, but for metabolic activity, PET. So with that, I'll conclude, and I thank you all for your attention. Thanks. Well, so MR, you might not want to use it for a patient who has a pacemaker. Pregnant, you wouldn't want to use, ionizing radiation. Yes, kids below the age of 18 are somewhere between 5 and 10 times more sensitive to radiation. So actually the regulations regarding kids, 10 times lower dose. So, but fortunately, the techniques like CT are coming dramatically down in dose. But still, if you have a choice, a non-ionizing technique is better for kids. So radiation is a big deal. Renal failure is a qualifier.
Where would the proton technology fit in?
Oh. So, are you talking about proton therapy? Yeah. So, outside of my domain of expertise, fortunately. But there are a lot of new technology, imaging technologies that are coming out, like hyperpolarized which we're doing naturally, but you won't find that in a routine imaging department. Also, optical imaging is really exploding, but again, not found in a radiology department yet. These are sort of growing technologies relying on fluorescence and that kind of thing. So it's very exciting. Go into radiology. Well, it's not as hard as it used to.
Okay, our next speaker is Frank Maldarelli, and he's an expert in AIDS. And, you know, the AIDS epidemic started some 30 years ago, and now we're faced with Ebola. But so far, Ebola has killed very few. AIDS has killed very many. So Frank got his PhD from the City University of New York, his MD from Mount Sinai School of Medicine. He did a residency in internal medicine at the Columbia Presbyterian Hospital. And then he came to NIH, and he's with the HIV Drug Resistance Program. His title is Retroviruses. Frank, always a pleasure to have you back.
Thank you. Thanks a lot, and good afternoon. I wanted to talk about retroviruses to this group, the NCI group, because as it turns out, it's our virus. It's certainly an NCI virus. I know we talk about HIV a great deal, and HIV certainly has a big presence in the NCI, but before HIV, there was probably as bigger presence of retrovirus study in NCI, and that's because these viruses, at least in animal systems and in some humans, also cause cancer. And so they're of interest for a number of different reasons, and I think it's a great topic, and I'm really thrilled to discuss it with you this afternoon. If you have questions, please just certainly interrupt. What I really wanted to talk about in general, this is just more of an outline, is to introduce these viruses, talk about their molecular biology and replication, because knowing that, we understand how many of the therapies have been developed for them. And then look at these retroviruses as they entered and emerged in human populations, and then talk about how that emergence and spread occurred, and then finally whether there are some lessons. And I think those lessons are not just for infectious disease people, but for researchers in general, because a lot of the things that, I don't know all the different things you're working on, maybe we can find out, but a lot of the research techniques that people use today have actually used retroviruses in one form or another, or at least have benefited by their stuff. So before we launch into HIV, let's start with the NCI virus, right? And that's human T-cell leukemia virus. And actually, this was the first retrovirus that was found in human populations to be a cause of disease. And here, numbers of places where this, I guess I could use this here. The numbers of places where this virus is either endemic or has traveled, and it's of interest, and we may come back to it later, that some of these places are sort of widely separated, either in the West Indies and then here in southern Japan, where the HTLV seropositivity can be as high as 30 to 50% in places like Okinawa, and then other places as well in Central Africa and in Central Asia. And I guess if you thought about it, you'd say, how many million people, all right, how many thousand people do you think are infected with this particular virus? It's like a guess. Okay, so this is a virology talk, so we talk in log terms, all right? 5 people, 50, 500, 500,000, 5 million, 50 million, 500 million. Take a guess. 500 people. Anybody else? Are there any other guesses? Okay, so from a virology standpoint, that's like I think about 4 logs off, maybe 5. 50, it's in the order of 30 million people. So this is one we don't talk about a lot, but it's one that infects about 30 million people. Now here, this is what's going on with HIV, and these numbers are probably downward. They're probably up closer to 40 million. The total number of infected people with HIV approximates, same order of magnitude. If you're a virologist, I can't tell them apart. HTLV and HIV are comparable in terms of their numbers. And obviously, they're distributed differently, and the risk that gives you HIV is distributed differently. So here, this is the region of Central Europe and an explosive epidemic that occurred in the former Soviet Union, where intravenous drug use accounted for about 2/3 of the infections. In contrast, at a place like Southeast Asia, commercial sex work accounts for the 40% most common cause of disease. And then you have places like the Caribbean where the risk distribution is fairly well distributed. And the one I'd like to point out are the ones you know, right? Commercial sex work, men who have sex with men, intravenous drug use, but then this one, other. In other words, a risk that is not being reported at the time, and likely because of the stigma associated with the disease that HIV causes, HIV/AIDS. So this stigma is real and continues to be a strong part of the epidemic. Knowing the molecular biology of this virus really gives you a great insight into a couple of things: the diseases it causes and life on Earth. So retroviruses are a group of RNA viruses that replicate by a DNA intermediate by reverse transcriptase. And I bet, because I'm looking at your faces, that you know this already. And it was discovered, or this phenomena was discovered in the middle 60s, and by 1970, the enzyme itself was identified, and it really changed the paradigm. And it was a new paradigm at the time. DNA to RNA to protein, right? Late 1940s, early 1950s, DNA was identified as the genetic material. Between the 1950s and the early 1960s, a lot of that work done here, it was found that, the RNA was synthesized in response to the DNA, and then that protein was made from those messenger RNAs. Well, this paradigm changed that. This said, I can take an RNA molecule and turn it into a DNA molecule, and now that's genetic information. And it's a way this particular virus replicates. And as it turns out, maybe, just maybe, this is how life on Earth persisted. So if you go out for a walk, and this is Great Seneca Park up on Clopper Road at Great Seneca Creek, and look at anything that moves or is alive or it has genetic material, all of them, every phylum, has in its genetic material a reverse transcriptase-like enzyme that functions to convert RNA into DNA. And as you all know, the life on Earth probably started as an RNA world. And that RNA world was put at great risk when the first products of their poop, as it were, was oxygen. So in an oxygen environment, I don't know, any of you work with RNA at all? You know that if you leave RNA out, you don't have any RNA anymore. And one of the principal reasons, besides the RNases that are present, is that RNA isn't stable in oxygen. And so back when we were an RNA world, RNA wasn't stable in oxygen either. And it was only those organisms, or at least the theory is it was only those organisms that could convert their genetic material into something a little bit more stable, DNA, that actually lasts. So one branch of retrovirology thinks that these viruses or this enzyme was like a Jedi Knight that essentially saved life on Earth by enabling organisms to convert their genetic material into a more stable structure. If we cone down now and look at these retroviruses as exogenous infectious agents, we find that there are about 7 different families of them. And the thing I really want to point out is that the ones that cause disease in humans are not the ones that are most closely related to each other. So here, lentiviruses, which cause HIV, there are two kinds, HIV 1 and 2. Their relatives, their closest relatives, are similar viruses in monkeys, in higher primates, in horses, in goats, and in sheep. And not their closest relatives are here as HTLV. So human T-cell leukemia virus infects humans. Its closest relative is a virus like it that infects cows, bovine leukemia virus. But here, other viruses that infect humans, also retroviruses, are not closely related. The ones that we're going to talk about for most of our discussion here are coned down again. Now, just looking at lentivirus relationships, we see the same phenomenon. So there are two viruses that cause disease in humans that are called human immunodeficiency viruses. And because we're a concrete bunch in this business, they're HIV-1 and HIV-2. And as you can see from this phylogenetic tree, HIV-1 and HIV-2 are both on the same tree, but they are not as closely related to each other as HIV-2 is most closely related to a virus that's found in monkeys, in a particular monkey, a sooty manga bee, that's given this designation, simian immunodeficiency virus, and coming from a sooty manga bee. That's its closest relative. Closest means it's 98% identical to the virus that's in sooty manga bee, where in contrast, HIV is probably only about 50% related, or 50% homologous to HIV-2, but it is quite related to a simian immunodeficiency virus that's found in chimpanzees. So immediately you see, from being a nerd and doing a lot of sequencing, that these two viruses are quite different from each other and perhaps have close relatives in other members of the animal kingdom that may give strong insight as to where this virus came. And as you can see here are a series of other viruses. Some that you may know if you have a cat, this is feline immunodeficiency virus, or if you have a horse, equine infectious anemia virus. Those are all related. But the ones that are most closely related to HIV-1 and 2 are in the primate community, one in chimpanzees and one in sooty manga bees. And this, for those of you who are scoring at home, this are retrovirus conventions. So this is what the genome looks like when it's when it comes from the virus. So this is an RNA molecule. As we'll see, there are two of them in each virion. And if you're interested in viruses and you looked at a bunch of them, this one in one way is actually unique among virus families. And that's that, viruses encode a lot of their own genes and so forth, and they have their own genes, their own enzymes, and they have their own promoters. Well, every virus family, except for the retroviruses, every virus family has a number of different promoters. HIV is unique in one respect, in that it has just a single promoter, and that single promoter is, oh, wait a minute, it's over here on the 3' end. And its polyase sequence, when you look at the RNA, is at the 5'. It's a molecular biology, depending upon how you want to look at this, either a dream or a nightmare. What is the polyase site doing up here? Well, we've seen some promoters being able to promote downstream, but this is a little bit unusual, especially when all of the coding sequences are. And the answer to this, and there's about 9 different genes, the answer to this comes if you look what happens when it's converted into a DNA molecule and integrated into the host genome, and that's here. That thing is called a provirus. So once it gets integrated, these ends become more complex, and now what was over here as what's called a U3, unique region in the 3' end, is now at both ends here. And U5, the unique region at the 5' end, is now at both the 5' end and the 3' end. And this R region, and if you're following, repeat, it directly repeats at both ends, is now at both ends. So once it integrates itself into the host genome, its promoter is at the 5' end, its polyase sequences is also at the 3' end, and it can synthesize all of these cistrons by using alternative splicing, as we'll see, with a major splice donor here, and then splice acceptors all along the way. So it can manage to express 9 different genes from a single promoter in this fashion. The names, I think if you've seen them before, if you read about them, have some unusual pronunciation. GAG, for instance, does not mean I am so sick of this. It means group antigen. In the old days, this is the way the serology could distinguish all of the different retrovirus families without being able to see it. POL stands for polymerase N, envelope. This is an envelope virus, as we'll see, and then on and on, just for your own a glossary for yourself. This is what the virion, an artist's depiction of the virion. It's enveloped. In other words, as it's budding from this infected cell, it acquires a cell membrane. That cell membrane has HIV-encoded molecules on the surface. These serve as both interacting with cell surface receptors and infusion. As we'll see, they're known as GP120 and GP41. And once again, if you think those people were who were so concrete to call things U3R and U5, they named the proteins by their apparent molecular size in polyacrylamide gels. That's the number, and either P for protein or GP for glycoprotein. So GP120 is a glycoprotein that has an apparent molecular size of 120,000 Daltons. 41 and P17, a protein of 17,000 molecular size, P24. And I guess the paradigm here is that there are a number of these proteins are encoded by HIV, and it includes structural proteins that actually make the core, enzymatic proteins that are going to reverse transcribe and integrate the viral genome, and 2 copies, count them, 2 of the HIV RNA, not complementary, but identical. So we're going to need both of these copies to replicate this gene. They're both plus-stranded, the same as the messenger. Knowing this replication cycle tells you a couple of things. First of all, it's so cool and interesting, I know. But the other thing it tells you is particular areas of replication where the virus has to cooperate with the cell, but may have a specific event that could distinguish it from a typical cellular process. And so working this kind of stuff out for a virologist, yes, it is a dream, I admit it. But it also identifies places in virus replication where you could target an inhibitory event or develop a drug. And you'll see at each one of the specific ones, a drug has been developed, is in use in the clinic, or is in development. So let's look at some of those. And the first one is, getting into this susceptible cell. And you might think that if you try and infect a cell, all you really have to do is interact with it. And then, I got a membrane, you got a membrane, the 2 membranes come together and pop. They're like two bubbles that you used to blow when you were kids, and maybe I still do, I admit it. The 2 of them come together and then they join up. Well, it turns out that's not exactly the case. And understanding how HIV replicated and some other viruses like influenza, a couple of really interesting and then important steps in virus replication were dissected. So attachment of that cell surface protein to a host cell that had a receptor molecule on it that will interact, that event is distinct. That attachment is distinct from actually getting inside the cell. So the first thing it does is attach, and it does that through this molecule GP120, and it has to interact with a host surface cell protein. And as you probably already know, one of those surface cell proteins is CD4. So CD4 is the receptor for HIV, but it's not enough to just interact with CD4. It has to interact with a co-receptor as well. And in the case, the principal ones for HIV are called CXCR4 or CCR5. They're molecules on the surface of lymphocytes and other cells like macrophages, brain microglial cells. But unless you have both of these, no matter how hard you try, you can't get that virus to it. So that's the attachment stage. And so it's possible to purely inhibit attachment. And that's a drug that's actually in the clinic now. It's called Ravirac, and it blocks the interaction of the HIV glycoprotein with the co-receptor CCR5. We actually don't have, after all these years, a good inhibitor of the HIV envelope CD4 interaction. But this CCR5 one works fairly well. It has to be used in combination or resistance. The next thing that happens is, as I say, it's not enough to just attach. You have to fuse those membranes. And you might think that's an easy affair, but it turns out to have a very high energy of activation. In fact, the virus has to literally push itself through the cell membrane. And it does that using GP41, also known as a fusion protein. So what happens is it interacts with the cell surface. Okay, fine. GP120 is already complexed with GP41. GP41, the changes in GP120 that occur cause conformational change in GP41, and that exposes, those conformational changes exposes a 6-helix bundle. And in this 6-helix bundle is buried an enormous amount of potential energy. They're like springs, and those 6 springs, once they've been exposed, force these two membranes together in a fusion event that is described here in great detail by this question mark, because we still don't really know exactly how that occurs. But the outcome of that rearrangement and forced fusion of these two membranes allows the HIV core into the host cell membrane. And so that's a pretty specific event, that fusion event. And here, if you look at GP41 at some of those bundles, you get the sense that they're all alpha helices. They really do look a little bit like springs. And looking at them head on, you can see as the viral membrane, as the cell membrane would see them forcing the two membranes together. And as a result, you think, well, you know, this might be a good time to develop a drug. And so this one, if you're thinking that might be easy, it turned out to be a lot of work, because what it turned out to be was a peptide. So they made a peptide, 36 amino acids, that fits in this groove and blocks the ability of these things to convert their potential energy into a kinetic energy. That turns out to be a tour de force from a pharmacologic standpoint. Merck had to build a new plant just to build this. This is the largest, as far as I know, 36-mer is the largest FDA-approved peptide commercially synthesized. So yeah, insulin's bigger, but you can purify insulin. This one is chemically synthesized. So once again, a great drug, FDA-approved, needs to be used in combination. What great drug? It has to be injected into the skin. You can't eat this one because it would be degraded in your stomach, but it does still work. It's a tough one to take, but it still remains effective. So working out those steps, we realized that viruses attach and fuse, and then the core gets inside the cytoplasm, and a funny event called uncoating happens. Now, I know what you're thinking. Yeah, what is this uncoating? It's like he's got to take his jacket off and expose this core structure. And within this core is an RNA, are these two RNA molecules. Well, I guess everybody thought, well, yeah, that probably has to happen. But, you know, how important can that be? It turns out a lot more important than a lot of people thought. In fact, cells can restrict virus infection at this stage. So even though you get in, you win. You attached, you fused, my core's inside, my core's going to start breaking apart. The cell has an innate immune mechanism that can restrict virus infection at this stage. Okay, fine. Some nerd found that in the laboratory. No, this one's more important than that. In fact, it's the reason why poor Terry has to listen to this lecture every year. Because if we had a better restriction mechanism in humans, this would be a virus infection of somebody else's primates and not us. This uncoating step, so here, you know, in a different artist, it attaches, it fuses, it gets in, here's the core, and then this core has to break open so that nucleoside triphosphates will, the enzymes can have nucleoside triphosphates in an intact core. That gets to be a difficult affair. So this step here turns out to require a gene product, or it can be restricted by a gene product called TRIM5-alpha. Has anybody ever heard of this thing before? Because if you had a good or if I had a good one, we wouldn't have to worry about this virus at all. TREM5 alpha is one of these innate immune molecules that surveys the cytoplasm for cores like this, and it can block the encoding step directly. I don't know how it does it. It's a pattern recognition thing. Don't ask me. It's immunology. But if this phenomenon is active, if TREM5 alpha can recognize this core, then this core doesn't get uncoded, and the virus gets stopped right there. So it requires the cell protein called TREM5 alpha, and it interacts with GAG. Here's an interesting little table. You have a TREM5 alpha, I have one, the monkeys have one, the chimps have one, all kinds of animals have these TREM5 alphas. And here, as I mentioned before, is a virus that infects humans. Here's one that infects the chimps, and here's one that infects the humans. Now, if you try in vitro to infect human cells with HIV, no problem. You try and do that with the chimp virus, no problem. You try and do that with an SIV monkey virus, no problem. How about some of the other things, the cells in this Excel table? Let's take, in the interest of time, we're going to skip the chimp. Let's take monkey cells, and we try to infect them with HIV. It's a no-go. They get blocked at this uncoding stage. How about the chimp? It's poor, it's crummy. SIV from a monkey, well, it infects monkeys, so it can get through this uncoding step. Well, comparing the monkey TREM5 alpha to the human TREM5 alpha was an interesting little affair. And they found just a few, maybe just one amino acid, critical amino acid difference between the monkey TREM5 alpha and the human. But that single difference allows HIV to infect human cells and is restricted in monkey cells. In other words, if this gene product is the gatekeeper, this gate is shut, this gate is open. And if you compare the TREM5 alphas of many different monkeys and species of monkeys, and then of humans and gorillas and so forth, you'll see that this gene is under intense selection. In other words, it's changing faster than just about any other gene that has been compared in this fashion. And the theory, I haven't been around, I'm old, but I'm not that old, is that over the last millions of years, we've had waves of these infections that have come into different species, cross species, and the TREM5 alphas have been one of those gatekeepers preventing infection. Or, look at it another way, if this virus infects everybody, only the people that have a good TREM5 alpha can exclude it. They're the ones that keep this species moving. So what some people think is the TREM5 alpha we have isn't just a crummy one because we're lazy and American and so forth. No, it has nothing to do with that. It's got a great TREM5 alpha, but it's for the last battle that we fought against an invading pandemic retrovirus. So humans that survive maybe have a TREM5 alpha that was great for before, but isn't so great for now. And so this, this is Charles de Gaulle had said, generals are always fighting the last war. And this perhaps is an example of that. It would have been great for the last one, but it doesn't stop this HIV. We could wait for evolution to take over. There's probably people out there who have TREM5 alphas that could resist HIV, but it would probably sacrifice most of the people on Earth because everybody else has got a susceptible. So at that stage, with one simple protein, we would have, you guys could be listening to a different kind of lecture. Okay, so that uncoding step, still don't know a lot about it. That might not be a bad place to develop a drug. Well, because these things are pattern recognition affairs, they tend to be more difficult to screen for drugs and more difficult to develop. So, okay, so let's go to something easy then. An enzyme, reverse transcriptase. Okay, so the cores come off. We have nucleoside triphosphates. We have these two RNA molecules, and we're going to convert them into a double-stranded DNA molecule. And now I am going to tell you something that you may or may not know. That for your whole lives, I'm looking around, all of you have carried around the three-dimensional X-ray crystallographic depiction of HIV reverse transcriptase. Don't look. It's your right hand. So DNA polymerases in general, and HIV reverse transcriptase in particular, looks like a right hand. It has 2 subunits, and it's got a palm domain right about here, and then a thumb domain, and then some figures as well. And let's see, a thumb domain up here, and then these fingers domains. And this is where the catalytic activity occurs, right on the surface of your thumb. And as you can imagine, you know, if you had an RNA molecule, this is my RNA molecule this afternoon, and every RNA molecule, when you replicate it, you need a template, you need a primer, and you need an enzyme. That'll be my right hand. And so it sits here across this groove. Nucleoside triphosphates are going to bind to these fingers domains. It's going to close over. It's going to incorporate that nucleoside triphosphate, give off the diphosphate, integrate, insert that nucleoside, and then move a little bit, and then do it again, and again, and again, and again, and again. And that's the how this catalytic activity takes place. Well, drugs that are going to inhibit that can inhibit it in 2 kinds of ways. One is to use a drug that looks like a nucleoside triphosphate, and we'll see some a little bit later, but you can add on to a so-called dideoxy. So any of you who are still doing sequencing, these are dideoxys that are used in the clinic as well. And the other thing you could do is disrupt the structure of the catalytic site using proteins or drugs that actually disrupt the structure around there. Both of those things are available for HIV. And in fact, a group of non-nucleoside reverse transcriptase inhibitors bind right around the active site, but not to the site itself, and change the structure enough that when the virus does become resistant, it changes the structure. So there's a great degree of back and forth in the polymerase reverse transcriptase. So the other thing, and this you may know, is that it has a number of enzymatic activities. I said I start out with an RNA molecule, 2 RNA molecules, and I'm going to end up with a double-stranded DNA molecule. And I'm going to do that only with reverse transcriptase, you know, with one hand tied behind my back. So it has to be an RNA-dependent DNA polymerase. That's its first biologic activity. But then, you know, the product of that first reverse transcription event, well, there's an RNA molecule, and now there's a DNA molecule. There's an RNA-DNA hybrid. I have to get rid of that RNA molecule if I'm going to make a DNA hybrid. So it carries with it another enzyme. Right in reverse transcriptase, another activity, it's called DNA-dependent, RNase H. And that cuts out the RNA from an RNA-DNA hybrid. It cuts almost all of it out. It leaves a few of them there that serve as the primers for the next round of DNA synthesis. So the next round now, there's no more RNA there, hardly. It's a DNA-dependent DNA polymerase. So RT, RNA-dependent DNA polymerase, DNA-dependent DNA polymerase. The one thing it can't do is an RNA virus. It can't make RNA, right? If you think about it, one more crazy thing about retroviruses is that it's an RNA virus that doesn't have an enzyme that makes RNA. It's going to rely on the cell. Its error rate is interesting. So has everybody heard this, that retroviruses have a very high error rate, and that's why there's a lot of genetic diversity, and that's why it's hard to make vaccines? Is that something that is only I think about? It can't keep me, you know, keeps me awake at night. You've probably heard this, right? Its error rate is about 1 to 4 mistakes every 100,000 bases syntheses. And I wish I made mistakes on that level. But if you think about it, the DNA polymerase that replicates our DNA is about 5 logs, 100,000 times more fatal. So the error rate is, it seems low, but for it, for a virus, it's about even. So look at any of the other viruses that replicate on flu, it's about the same, right? Any of the other, anything that you may be having right now, a cold virus, about 4 out of 100,000. Anything that has to replicate RNA, and in this case, we're going to be using RNA into DNA, but anything that has to replicate RNA has a substantial error. There can be recombination during replication as well, so there's a a lot of ways to make genetic diversity. So replication occurs, as we'll see, completes in about a day. So the combination of a rapid replication rate, generation being a day, generation for people about 33 years, they're a little bit different. A replication rate that's both rapid and error-prone gives a very highly genetically diverse population of virus in an individual. And I think that implies this piece that we're going to look, and as you probably know, one of the biggest problems in treating HIV is the development of resistance. Well, what I'm going to argue is that most of those resistance mutations are present even before the patient takes the first pill. They're a consequence of this rapid and error-prone replication cycle. And I'm going to argue that that rapid and error-prone replication rate is actually not just some leftover mistake from saving the world, right? It could have been a great enzyme. It just never evolved into a good one. In fact, I think it's a pathogenic determinant for virus replication. Think about it this way. Each round of replication is going to generate a number of different mutants, about one per every genome synthesized. Some of those mutants are going to be dead. A lot of them are going to be dead. Most of them, but some of them aren't. Some of them are going to be exactly what they were before, and they'll be fine. Some of them will be slightly different. Many of them will be tolerated in a diverse, genetically diverse population. And that genetically diverse population can then respond very quickly and dynamically to a selective pressure. Drug selection pressure, sure. But in this case, in the primary case, it was probably an immune selective pressure. Consider this example over here. Whereas I know you don't have to take out your genetic code because you guys are so smart, you memorized it all. There's a particular position in the reverse transcriptase 151. And normally, most of the time, 99% of the time or more, it's a glutamine or CAG. Change one nucleotide from CAG to CTG, and now it's no longer a glutamine, but a leucine. Well, okay, not the greatest virus, but I'll tolerate it. CAG to AAG, we go from glutamine to lysine. Okay, once again, okay, make one more change and go from either AAG or CTG to ATG. As everybody knows, that's methionine. You get this population. So now I have a population of 4 different variants. This one is the vast majority, and these 3 are less likely. But in the presence of a drug, any of the nucleoside reverse transcriptase inhibitors, all 3 of these are tremendously inhibited, and this one is not. So this population, before they ever take the first pill, already has a mutation that confers resistance to that particular set of drugs. And so if you think about it in terms of drugs, fine, the virus didn't evolve that for that purpose. Probably more in line with an immune response. So the same thing happens at CTL sites, where a number of different ones are tolerated, and the minute a response occurs, the virus already has something that can expand quickly. So once again, this rapid and error-prone synthesis is likely to be one of the more important pathogenic determinants for the virus. Once that reverse transcription takes place, then the double-stranded DNA now is transported to the nucleus and integrated into the host gene using a viral gene product called integrase. And at this point, the enemy is within. Integrase is an absolutely essential step at virus replication. So once you hear as an essential step, the next thing you know, well, maybe I can make a drug for that. It's an enzyme. We can screen quickly. And integrase inhibitors are really the most important drugs developed in the last 15 years for HIV. There are 3 of them now on the market. They're extraordinarily good, and they block the ability of the double-stranded DNA to be integrated into the host genome. And this, again, is, once again, for those of you who are going to go into or thinking about the pharmaceutical industry, this was one of those times where they developed a bunch of really good drugs that looked great on paper. We had them here at NIH. They were relatives of cancer drugs, and they were kind of toxic. So they said, well, we're not going to give up. We're going to invent a new assay, and we're going to screen a completely new set of drugs. They did that. They found a great set of drugs that will inhibit integration. It was terrific. You know, everybody was poised to develop these drugs, and they found out they were pharmacologically poorly absorbed, and they were no longer able to be developed for that reason. So they had gotten all that way. Merck would not give up. They went back to the drawing board, and they developed a whole new set of drugs based on an assay they developed, and they finally got these drugs developed and into the clinic. So not giving up is an essential characteristic if you're going into drug development. Once the provirus is integrated into the host genome, as I say, the enemy is within, and now it relies on the host. It's not going to take over this host cell. It's not going to shut off any host cell synthesis. It's pretty bright about things. It's going to let everything just happen the way it happened before. It's going to use RNA polymerase to generate its RNA. It's going to use RNA processing equipment that the cell has, alternatively splicing many of its RNAs to produce all kinds of messengers that will translate into the gene products that it needs to assemble at the cell surface to make a virion. All of the processes that it uses in this post-integration events are cellular processes. So it doesn't shut anything off or cancel the cell. Yes, the cell is triggered for death, but not until it makes a number of these virions. And at the end of this process here, virus particles start to bud off of the cell, getting a membrane that they get from the host cell membrane. And once again, here's another step that is essential for virus replication and is a site to develop new drugs. So the virus, when it comes out, I think you can see the difference between these here and these here, and this one here. Here, anybody familiar with electron microscopy or does any electron microscopy? Ever look at electron micrographs? Is this the very first one? Okay, fine. Now I got it. So you know that you have to slice these really thin, and you have to stain. And you stain them with things like urinal acetate, osmium tetroxide, or lead. You guys know the chemistry better than I do. All of those things are highly positively charged, heavy molecules, lead, osmium, etc., right? So you can imagine if you pour something like that on a slide like this, you got all these positive charges, they're going to go looking for negative charges. And the first negative charge, I think, and the most concentrated one, are the ones that are in the nucleic acids. Yes, proteins have negative charges on them too, but you think about it. You know, nucleic acids have a phosphate every time it's got a base, 9707 nucleotides. That's a lot of phosphate. So whenever you see something like this, at least of this virus, wherever this is, with the dark stuff, the osmium, the thing that's deflecting the electron beam, is where the nucleic acid is. So yeah, this is a virion. And I know it's a virion because its nucleic acid is being stained on, plastered up the inside of this virion particle. Well, over here, it's concentrated in this core-like structure. So something happened between here and here, and that thing that happened is known as maturation. And I'm droning on about it because it requires an essential step in virus replication mediated by HIV protease, which converts this stuff into this stuff by digesting a polyprotein precursor into its products. So the important thing here is these are immature particles, these are mature particles. These are not infectious, and these are infectious. So unless HIV protease digests those precursors, no matter how many of these you have, they are not going to initiate the next round of replication. For once, we get to be the ones to grab victory from the jaws of defeat if we can inhibit protease. So protease inhibitors, about 15, close to 20 years ago now, became some of the critical components of combination antibiotics. And so knowing those steps antiretroviral therapy was developed, as we'll see what those drugs are. But considering that replication is rapid and error-prone, you can't use just one. We have to use them in combination. Well, I wanted to take just a moment to look at where these viruses actually came from, how they emerged in human populations. You saw this before, that the biggest clue to this was seen in its viral sequences, that HIV-1 was most closely related to chimps, HIV-2 to the sooty manga bee, and if you look here in Africa, well, that's a place where those monkeys and humans have cohabitated for tens of thousands of years. And so an experiment that survey that was done was to see whether or not we could find the relatives of HIV and HIV-1 and HIV-2 in these monkey or chimp populations. And so I know you guys work extraordinarily hard. I don't know, you postdocs or ERDAs or staff scientists, I think people at NIH work really hard. And I think we can identify with the people who did this stuff. So what they did was to go out and get samples from chimpanzees to see if they had any of this virus-like in them, how prevalent was it, and was it anything like the human virus. But it's harder to get samples from a chimp because they're protected, and you can't bleed them. So the people who ran these protected forests were very happy to tell the postdocs where the chimps slept at night. And the postdocs could collect a couple of things. They could collect urine, and they could collect poop from these animals. And they knew where they slept, they knew where they peed in the morning, and they knew where they pooped. So they could collect that stuff and send it back to the lab and see if they could find remnants or pieces of retroviruses in those bodily fluids. And in fact, they could. So you can extract chimp poop and find sequences of SIV that are actually quite similar to HIV. The same with urine. And you'd say, well, come on, you know, all urine kind of looks alike. And poop, give me a break. So what they did was actually were also able to sequence chimp sequences out of that poop and out of that urine to prove that this urine was not just anybody's urine. This was urine from a chimpanzee. And here is a phylogenetic tree, a very hard-fought phylogenetic tree that shows that the HIV sequences that are found in humans are very similar to these chimp sequences that were identified in a preserve here in West Africa, also one in Central Africa. And interestingly, and I didn't know this, there are 4 different species of chimpanzee. They're all pan-troglodytes, and either virus virulorosus, troglodytes, or Schweinfurthii. And this population, which probably left the main herd at least 10,000 years ago, does not have detectable HIV. So monkeys, these chimps don't like to cross rivers. I guess some did, and they established this colony over here. These animals don't have HIV. These do. And they probably got it through getting it from lower monkeys. So how did it get into people? Well, there's this bushmeat trade that usually starts with a poacher. Here's one with a sooty manga that he brings to a chop house. It's sort of like a butcher who makes all different cuts of meat, and those are sold at a bushmeat market. It's a pretty common thing in Africa, and then it goes to homes and to restaurants. And you'd say, well, you know, if it's a pretty common thing and people have been eating these animals for thousands of years, how come we got so lucky to get this epidemic now? And I don't have an answer. There are some theories, but I think it is possibly true that this kind of zoonotic event has been happening for the last 10,000 or more years, and it just hasn't flamed up into this terrible pandemic that we've experienced. If you look at the viruses that are infecting humans, all the different HIVs, and do more sophisticated phylogenetic analyses, you can find that you can trace them back to a virus that, yeah, probably with some, you know, play at the wheel, was probably as early as 1911, maybe as late as 1945. Newer data suggests maybe the 1890s is the oldest estimated virus in humans. But what was going on at that time? We know about, we know about the ways that we can get HIV by blood and body fluid transmission, mother to child, perhaps blood transfusion. But then there are these other ways that the virus gets transmitted in populations. They're political, they're economic, they're multiple epidemics. So here's a map of Africa, and this is from, I think, the early 1800s, and then here around the time of World War I, the early 1900s, and then here in the early 1960s as these countries gained their independence. And I think even, you know, a poor historian like me could see that there's been a lot of change over this time. And that's included political upheaval, population movement, potential for malnutrition. And a malnourished human is relatively immunosuppressed. So perhaps they were more susceptible, those people were more susceptible to getting infection. And that's why more people might have got infected from a few zoonotic events. The population increased in certain places in Africa fairly rapidly in the 1920s and 30s. And this is Kinshasa in Central Africa, and its population in billions, which went up quite dramatically between 1920 and 1930. And that's thought to be an important determinant of transmission. And then people became more connected. And so this is the Trans-Africa Highway that has been bringing goods back and forth across Africa. And these, just like the New Jersey Turnpike, the Trans-Africa Highway is punctuated with rest stops. The rest stops have food and hotels, and people have traced some spread of HIV across these highways. So the result of that has been the transmission of HIV throughout Africa. But how did it get to the United States? And I think if you look at this alphabet soup, these letters are designating different clades or viral subtypes of HIV. And so you see in Africa, there are many, many of them as they diversified. But then if you look at other places, well, the United States, it has a lot of just one subtype. 95% are B, and the same is probably true in Europe, even more so in Australia, and a couple of subtypes here. How did that happen? And so that's the result of a viral fouling effect, a group of viruses that entered a new geographic area. And I think some very sophisticated phylogenetics has suggested that that transmission, although I think I have it, that transmission was thought to be urban legend, great story, probably not true, by an airline student. Maybe you've heard this story. His name was Gaetan Dugas, who thought he was patient zero for the United States. And as it turned out, I think the good phylogenetics suggested that more likely that the virus made a stop in the Caribbean and Haiti before coming to the United States. So you could find viruses in Haiti that are older than any of the ones in the United States and younger than any of the ones in Central Africa. So it's most likely that that transmission was much more complex than a single airline. This is how the epidemic emerged in the United States. This is 1983, and each one of these dots represents about 30 cases. And so 1980, this is data from the CDC, 1983. The case load goes up tenfold by 1985, still centered largely in metropolitan areas. In 4 years, it goes up tenfold again. Again, we're all virologists here, showing that the 95 corridor is getting quite well populated. And here, fivefold more increase by 1995, and the caseload now in the United States somewhere around 1 to 2 million. But you can see the epidemic exploding in that period from 1983 up to about 1995, and then leveling off. A much more mature epidemic depicted here in its presence. And so now, if you look at new cases of HIV, they're still occurring in men who have sex with men, but a third of them are now occurring in heterosexuals, and the IBDU, intravenous drug user, is still sort of a stable 10 to 12%. But some interesting trends in the epidemic. African Americans now exceed whites as the most common patients with HIV. Those of you who've been living in the district know that the District of Columbia has the highest prevalence of HIV in the entire country. Fastest growing group of people getting HIV in the District of Columbia is African American. Women in general comprise about 25% of all the cases, and it's an aging epidemic. By next year, 50% of the people living with HIV will likely be over 50%. This was a poster on a bus in 1988. If you get the AIDS virus now, you and your license could expire at the same time. No cure, no treatment. Over the course of the next 7 or 8 years, people took these events that we've talked about, all the steps in replication where there's an emphasis on the virus input over the cellular input, attachment, fusion, reverse transcription, integration, here, maturation, and developed a whole series of drugs, many of which are now FDA approved, but still identified a number of targets that new drug development can be used. So over the course of now from 1987 all the way up to 2015, these are all the drugs that we have for HIV, color-coded according to their mechanism of action: reverse transcriptase inhibitors, protease inhibitors, co-receptor inhibitors, integrase, and fusion. I think there are a few lessons. I don't know, right? The easy ones, right? Viruses are bad and should be avoided. But this one probably helped keep life on Earth. Not HIV, but the retrovirus group in general. And maybe, right, they saved us from the last epidemic. So there was probably a wave of virus that spread through humans. We evolved the TRIM5 alpha that prevented us from getting that virus. And it, you know, we didn't luck out, and it didn't prevent this. But understanding that can help us go after the next epidemic. Epidemics obviously are not just single events. They're complex affairs. They mature and they evolve, and understanding how they do that will help us control what we have now. So I think I'll stop there and see if there are any questions. You guys all knew this. I was aware. Just reinforce. Very slowly. So I think there was a very strong push, obviously, to develop antiretrovirals. And I guess I should have mentioned that the very first one, AZT, came off a shelf at the NCI. So the very first drugs were developed right here at NIH. But what was also clear was that some of these things were going to be fairly expensive. You heard that crazy story about the T20 fusion inhibitor. Well, by the time the drug companies make these things, they sort of want to hold on to them. And so there was a very awkward time when we had antiretrovirals and the developing world, where maybe 28 million of all the 35 million infected people were not getting those antiretrovirals. So there was a huge push by a couple of forces. I can't say we were missing at the table. So in the early 2000s, PEPFAR, the President's Emergency Fund for AIDS Research, was developed. This was $5 billion that George Bush essentially appropriated for this work. And I want to point out that I think that was a really, really good thing. You may say what you want about that president, and I've said a lot of things, but that was a good thing. And I'm also going to say that I think it was driven by people at NIH. And so the two people, I think, or two of the people who were very strongly involved in the President's Emergency Fund were Anthony Fauci. You may have seen him on television lately talking about Ebola. If you haven't turned on the television. So yeah, there he is. And I'm just coming from a meeting over here in Natcher. He was at that one too. And for those of you who work in Building 10, he makes rounds on the NIID floor at least once a week. And he has been doing that certainly as long as I've. So he's a force of nature. He saw that appropriately focused money could actually do something. And those of you who are fellows here, one of his fellows, Mark Dybel, became the first AIDS ambassador. So that made that, as far as I'm concerned, those three things made that, four things made that thing work. Fauci, Dybel, Bush, $5 billion. So it sounded to me like a drop in a bucket at the time, but it really primed the pump. And it also prompted other people to do other things. So the Gates Foundation has been funneling money there. The Clinton Initiative, you know what that guy's like. He's a great politician, and he talked people into bringing money to places like sub-Saharan Africa. And then, meanwhile, in another part of the world, places like India decided, well, we can make these drugs too. And they challenged the patents. And that was, maybe a little bit more friction than anybody wanted, but those drugs became available in places like that as well. So I think the United States, the WHO, and certainly a number of other companies pushed to have those drugs be made available. And it sets up a really interesting, for nerds like us, it's terrific. It also sets up an interesting little experiment. What happens in the United States where we have, you saw those graphs, how fast the thing evolved. By the time we had good medicine, 95, we pretty much had a stable endemic HIV. All right, we're going to drop drugs into that. Okay, fine. And we're going to get drug resistance because people are not going to take them adherently because these pills stink. And they're great. They stink. They're hard to take. Got to take them a couple of times. Things have gotten much, much better. But what happens in a developing country where people there are just as desperate for those medications, but the epidemic hasn't leveled? It's still going up. Now I'm putting medications into an active epidemic. And there's still real questions as to whether you'll get a lot more resistance or you'll get about the same. Some of the data suggests that it's relatively stable. If you look at all the people who are coming in newly infected with HIV in a major city in the United States, about 15% of them already have a drug resistance mutation, even though they've never taken a pill. And that's because the people they got it from had drug resistance. And maybe those numbers are a little bit higher in some places or lower in others in Africa, but they're about 15%. And it depends on the drug as well. I'm giving you a little space. But I do think the NIH had a lot to say about these pills, and I think they had a lot to say about how they got the substance. Yeah. They can. And yes, they can. And in fact, that's a really important point. Maybe I should revise the lecture to reflect that. Over the course of about the last 5 years, a number of really good studies have been done to show that if you give the antiretrovirals to the uninfected part, you can prevent transmission to the uninfected part. It's called pre-exposure prophylaxis, or PrEP. It doesn't work if you don't take the pills, but if you do, it actually works. Post-exposure prophylaxis also works. I think it's a little bit more tenuous. We obviously use it in the clinical center all the time when somebody has a needle stick exposure or something else, and it's very, very effective in preventing HIV. But you bring up, I think, a really important point in the epidemiology of this virus isn't just trying to figure out what its prevalence is, but in patient education to make sure that when patients are identified, they understand that that medications are important and need to be taken adherently if people are going to remain suppressed. So worldwide, I think about 30% of people develop drug resistance in one form or another. It's not. I don't think in sub-Saharan Africa, because I think it's more immediate. You know, and I count myself as a young people. Young people today do see HIV differently than they saw it initially. And I think I see a lot of people when they're newly diagnosed, and they say, oh, well, this is a controllable disease. This is a treatable affair. We don't have to worry as much. And I saw these same people 30 years ago when there were absolutely no pills. Those people were absolutely desperate. And they saw all of their friends and dying from it. I think Africa is a little bit like that. So it's still very immediate over there. They have a lot of orphans. And so I think the education, yes, has to be extensive, but I don't think it's a hard sell. Thank you very much. Thanks for sticking around on a cold and rainy day.